Welcome to our first online lesson for Unit 7. In this unit, we're going to be talking about cell division. We're going to be talking about the structure of chromosomes during that cell division process and what happens, how do organisms grow and develop and repair their cells. So here we can see our baby eagles. And a question scientists had was what happens to our cells when organisms develop into adulthood. So as these baby eagles become an adult, what is happening to their cells? And this is true of all living organisms, plants and animals. We are all made of cells. If we are multicellular, what's happening during that process? Are cells just becoming larger over time as we grow? Or do cells divide and make more new cells as organisms go into adulthood? So that was the question scientists had. What is happening to our cells in multicellular organisms? And so you can see here, from looking at a developing embryo, that cells actually divide as we grow and develop. And this happens in embryos and also into adulthood. Our cells continue to divide when we need to repair or grow. The two main reasons that a cell divides rather than continuing to grow are one, because the larger a cell becomes, the less efficient it is at moving nutrients and waste materials across the cell membrane. So for example, last semester we talked about the process that occurs in a cell, in a little organelle, and there's many of these throughout this cell, called the mitochondria. So if you remember from last semester, the mitochondria required glucose, and oxygen for this process to occur of cellular respiration. And it released our waste products of water and carbon dioxide. So as the cell, if it was increasing in size, it would get harder for the cell to obtain the nutrients that it needed to continue to make energy for cell functions, and it would also have more waste that it would need to get across the cell membrane, which make it more difficult the larger the cell is. The second reason why a cell would divide rather than continuing to grow is the increased demand it has on its own DNA. So DNA is our genetic material, and as a cell grows, the amount of genetic material is not doubling or increasing as well. And so that produces a strain for this genetic material because it is what makes all of the proteins for our cells. So as the cell gets bigger, it wouldn't be able to keep up with everything the cell needs. So it's easier for our cells to divide rather than to just get larger over time. So if you remember, we just learned from this unit that our DNA structure is a double helix. And these double helices can be up to 2 meters long. And in eukaryotes, DNA is found in the nucleus. But in the nucleus, it's not found in this linear structure. Our chromosomes are actually more compact, or tightly coiled in the nucleus. So DNA codes for all of our genetic information, and it is in all living organisms. So it has to be tightly packed or condensed into structures that we call chromosomes. And the reason for this is because our DNA can be up to 2 meters long. And in humans, we have 46 chromosomes. So there's 46 of these inside of a nucleus. So how does that fit into a cell and into the nucleus of a cell. How do we get those to fit? And that is where this DNA packing comes into place. And so our DNA becomes chromosome structures. Before we get into how DNA is packed into chromosomes, we're going to talk about some of the differences between DNA in prokaryotes and DNA in eukaryotes. So in prokaryotes, which for example were bacteria, their DNA is circular and it's free floating in the cytoplasm. And so circular DNA, it's still double-stranded, still made of nucleotides, but it's just a different structure than in a eukaryote, also referred to as plasmids. And they only have one chromosome, and again, they can have also plasmids in there. Eukaryotes, which are cells found in plant and animals, we have linear DNA strands, and DNA is found in the nucleus of our cells. And we can have several chromosomes, more than one. For example, humans have 46 chromosomes in their skin or muscle cells. Fruit flies have 8 chromosomes in a wing cell in one of their body cells. And bananas have 33 to 34 chromosomes because our bananas now have been genetically modified to look the way they do today. So these chromosomes can range. So chromosomes, how do we get all 46 of those chromosomes in humans to fit inside of the nucleus? So our DNA is a double helix. Structure. But that double helix is actually going to be coiled around a histone protein. Histone proteins are found in a complex of 8. So think of this as like a cube-looking structure. So our histone proteins help to supercoil our DNA. So if I start to draw our DNA more as linear, these histone proteins are going to be the structures that our DNA is going to wind around and begin to condense. So our DNA will start to coil around these histone proteins and form what is called chromatin. And this is condensed DNA. And it starts to coil around. So it's still in this double helix structure, but it's condensing around these histone proteins. The complex of DNA wrapped around a histone protein is called a nucleosome. And so most of the time, our DNA in the cell, in the nucleus, is in this chromatin structure. But right before division, our DNA begins to tightly coil even more into what's called supercoiling. And tightly coil even more into this chromosome structure that we see in our textbooks. And chromosomes are just supercoiled DNA. And again, this occurs right before our cells are about to divide. So our DNA, which is in the structure of chromatin, supercoils even more to form a chromosome. We learned that before our cells divide, DNA is replicated. So our chromosomes, and what you'll see in your textbook, are often drawn in these X-like structures. Because after replication, we have 2 copies of every DNA strand. And again, our DNA is tightly coiled around. This is after replication. We would have a chromosome that has 2 identical halves. Both of these are considered one chromosome. So again, our chromatin is going to supercoil to form this chromosome structure. Again, going into chromosome structure. So what happens to our chromatin right before cell division is it's going to supercoil and condense. So chromosomes are defined as having a centromere. And the centromere is a protein at the center of our supercoiled DNA. Centromere kind of sounds like center. So again, our DNA is supercoiled. This is just one strand. So our centromere is here at the center of our chromosome. So we're defined as having one centromere. This is before replication. After replication, we're still going to have one centromere. But DNA replication makes 2 identical copies of DNA. So you're going to have 2 identical copies attached at one centromere. And again, our supercoiled DNA. So this is after replication. But chromosomes are counted by the number of centromeres. So both of these are still considered to be one chromosome, but this is a single chromosome, and this is a replicated chromosome. And this occurs after DNA replication. But again, still, both of these are only one chromosome. So our replicated chromosome